Record the lesson. So earlier we discussed how kidneys and lungs are these are two organs that are responsible for controlling water content in the body, right? By the way, Hiba, when I'm expecting your result, your test, like I'm waiting for your answer sheet. Okay, hoping to hear back from you. Now, let's begin. Very good. Now, so we discussed that maintaining water potential of the body, two organs are involved: lungs and kidneys. The lungs mainly excrete carbon dioxide from the air that we have exhaled, like inhaled, and it exhaled carbon dioxide. Now, the lungs will excrete carbon dioxide, and we know that this carbon dioxide is actually a product of respiration. And which form of respiration particularly? Aerobic respiration, right? Then kidneys are responsible for the excretion of excess water. Make sure you write excess water, salts and urea. The kidney excrete water, salts and urea, and this urea is basically formed in the liver. Okay, urea is formed in liver, however it is excreted by the kidneys. So you should know that urea is formed in liver. This comes in MCQs, so be very careful. However, urea is excreted out from the kidney. Now, how urea is produced? Urea has many components, and one of them is amino acids. The extra amino acids we take in protein are removed in urea along with water. Urea contains water, excess amino acids, excess water, excess mineral ions, and what else? Creatinine. Creatinine is a compound made by our muscle cells, so you just have to know the name, that's it. Any confusion? Okay. Now we have the second subtopic here is formation of urea. Okay, we'll see how urea is formed. We'll give the heading formation of urea. The very first step in formation of urea is deamination. This step 1 is deamination. Now, first of all, you should know that excess, for example, if a person had 50 grams of chicken in a day, chicken is a pure protein, right? Obviously, the body would require only 20 grams of it. So 30% of it will be wasted as excess amino acids. From the gut, it will be absorbed as amino acids, and these amino acids will then move to the kidney and then the liver. These amino acids will move to liver actually, and from the liver, urea is produced and then it moves to the kidneys, from where it is excreted out. So this happens in deamination, basically happens in liver. Okay. Now, deamination, you know about amino acids, right? They are the subunits of proteins. So deamination itself is a process of breaking down excess proteins. Here, for deamination, we need proteins, we need enzymes. To digest, to deamine a protein, an amino acid, we need enzymes. Can anyone tell me the name of the elements present in amino acids? Any significant element? Okay, seeing no reply. The elements present in amino acids are nitrogen, carbon, and hydrogen. These are three major elements present in amino acids. Basically, the nitrogen part of your amino acid is turned into ammonia, and you should know that ammonia is really toxic for our body cells. When ammonia is produced, ammonia is a toxic gas, so it must be excreted out urgently with immediate basis. Right? Now I'm going to show you the structure of amino acid. Any confusion so far? Okay. Now this is amino acid. A single amino acid has amino group. Amino group means NH2. Then it has an R group. Basically, R group can be anything. It can be CH3, it can be sulfate, it can be any other compound or element. It differs from one amino acid to another amino acid. And then it has a carboxylic group. You must have studied carboxylic groups in organic chemistry yet. So this one is your carboxylic acid. Then we have a single R group, and then we have NH2. This is your amine group. This is your amino group. Do you know how many amino acids are there in our body? How many amino acids are there? No, there are 20 amino acids. And using this 20 amino acids, thousands of proteins are made. There are in total how many amino acids in body? 20 amino acids. Now, the enzymes will act on this amino acid, and it will break it down into its subsequent part. Now, this carbon containing part, this part and this part forms, like splits into glycogen. It forms glycogen, and in the liver it is stored in glycogenic form. Fine. The other part containing the nitrogen is converted into ammonia, which is highly toxic. It is converted into ammonia. This is why we say that it is deamination, because we are removing the amino group. Right? Now this ammonia produced is highly toxic. As it is toxic, so it is immediately converted into urea, which can be transported around the body via the blood supply this urea to the kidneys. Okay, the urea travels through the blood to our kidneys. Any confusion? Any questions? Okay, thank you. Now, let's discuss the structure and function of kidneys. So you should know that all the things, all the facts about amino acids and its deamination. Now let's move ahead. Now, this is the track pathway for our kidneys. This is the track for urea. Now, I want you to open up your mics and then speak, because now I won't be able to see the messages. Okay, you can use your mic, please. Now, we have two kidneys, and kidneys are responsible to control the water content of the body. The kidney controls water content of body, along with the concentrations of some ions, for suppose potassium or sodium ions, because these two ions are usually present in the fluids of the body. Now, each kidney contains a lot of capillaries. Can you see this aorta is bringing the blood via it joins the renal artery, and then renal artery supply the blood to the kidney. To which kidney? To the right kidney. And then renal vein is taking the blood back from the kidney to the heart. So let me repeat it. The kidney receives blood via, it gets blood via which artery? Renal artery. And this blood will obviously have high amount of urea, because urea is supplied by the kidneys, by the liver to the kidneys, right? Now, when the blood passes through the kidney, there is a pressure buildup inside the kidneys, and inside the kidneys we have a lot of filters. Fine. When mass flow of blood takes place inside the kidney, only small molecules passes through the kidneys, from the capillaries to the kidney. Okay, let me repeat it. See, don't need to worry. We have small molecules. When the blood passed through the renal artery to the capillaries surrounding the kidneys, only small molecules pass through the pores, and the larger molecules are stopped. The larger molecules cannot pass through. Can you give an example of large molecule?
Like salt.
Salt perhaps is a very small molecule. It's a tiny substance. Large
I said
molecule. Okay. Large molecule can be glucose. It can be an amino acid. Right? Any query? Only small molecules will be able to penetrate from this blood into the capillary. This step is known as filtration. The first step in kidney is known as filtration. In filtration, small molecules leaks out from the kidney, from the blood to the capillaries. Now, these substances are forced out, including glucose, urea, water, and ions. All the things are dissolved. Now, this liquid, including glucose, amino acids, water, urea, they all form a substance known as filtrate. We call it filtrate. So, filtrate is produced in filtration in the first part of the process, where it includes glucose, amino acid, water, and mineral ions. Right? Now, for suppose red blood cells. Red blood cells are significantly large. So red blood cells cannot escape to the capillaries, and hence the red blood cells or proteins will remain in the blood plasma. They'll remain in blood plasma. Any confusion?
No.
Okay. Now, the next step after filtration is reabsorption. The second step is reabsorption. In reabsorption, useful substances are reabsorbed to the blood. Substances are reabsorbed into the blood. Now, can you name any useful substance for body which is present in our filtrate?
Water,
Water, if it's not in excess, and
glucose,
glucose. Yes, glucose is a substrate, so it is a must for cells. Then, what else? Sometimes mineral ions are also needed. So mineral ions are also reabsorbed. This is known as this process is known as selective reabsorption, because the only selective molecules are reabsorbed while the others are excreted in urea. So anything that is not absorbed, for suppose excess water or excess mineral ions, they'll form a fluid, and this filtrate is actually excreted out. This filtrate becomes urea, and then it is excreted out by our urethra. Any questions? Okay, now look at this diagram. So kidneys, once the urea is formed inside the kidney, the urea travels through this tube. This tube is known as ureter. It is known as ureter, and then it joins to an organ, to a muscular bag which is known as bladder. Inside this bladder, urine is stored, and then the stored urine, after a limited time, it passes to the through urethra outside our body. Okay. Any questions? Hiba, Pravalika.
No. No.
Okay. Now, be very careful to describe clearly where substances are moving from and into the kidney. For suppose the glucose moves from our filtrate into the bloodstream because it must be reabsorbed. Our body requires it. Whereas excess water will be removed. It won't be reabsorbed. Now, let's discuss how homeostasis maintains the constant concentration of water potential. So for that we need to understand a diagram. Shall I continue?
Yeah.
Okay. Now, for suppose on a hot day when most of the water is lost, via other processes such as respiration and sweating or in tears, the kidney are responsible for maintaining the water potential gradient. However, as we are discussing hormones, so here pituitary gland is involved. The pituitary gland is sensitive to changes in water potential. And where is pituitary gland? Pituitary gland is located in brain, right? Pituitary gland is present in brain. This region, this gland is sensitive to water potential changes. Now, pituitary gland actually secretes a hormone known as ADH. Do you know the full form of ADH? Anyone? Antidiuretic hormone. The function of ADH, see, the quantity of ADH determines how much water would be reabsorbed. So if large amount of ADH is released by our pituitary gland, then it will lead to more absorption of water from the filtrate. Fine. High ADH, so more water is reabsorbed. Any questions? So let's discuss two factors when ADH is in high quantity and when ADH is released in low quantities. So for suppose if the water potential, the water content of the blood is too high, then the pituitary gland would secrete less antidiuretic hormone. Pituitary gland secretes less hormone. It releases less hormone, and when less hormone is released, it will lead to less water being reabsorbed in the tubules of the kidney. Basically, it maintains permeability of the capillaries present in your kidneys, the tubules present in your kidneys. It maintains permeability of tubules of kidneys, right? Any questions?
No.
Okay. Now, when water content of the blood is high, what will happen? Less ADH would be released by your pituitary gland, and as a result, less water will be reabsorbed in the tubules of the kidney. That means this is how the water potential would be monitored. Any confusion? Hiba? Pravalika?
No.
Okay. For suppose if the ADH concentration is low, as like if the water potential is low, is too low. If the water potential concentration is too low, then the blood would become concentrated. Concentrated means it will have less water. So pituitary gland will get activated and pituitary gland will secrete which hormone? ADH in high quantity, which leads to less water being reabsorbed in the tubules. So now let me correct the arrowheads. If it is too high, less ADH is released, and if it is too low, the water potential is too low, then it will release more ADH. Any confusion? This is an example of negative feedback. Negative feedback is actually a corrective mechanism. It corrects, it helps to bring back the set positions to its point. For example, if let's suppose the glucose concentration should be 120 mmg. So anything done to bring back the glucose concentration at this point is known as negative feedback. And we have already discussed how body regulates the glucose concentration in both the cases, right? Now, for suppose conditions in the body change from set point. The change is detected by which organ? By pituitary gland, right? Then corrective mechanism is activated where permeability of kidney tubule is monitored, like is changed. Tubule is altered. Then conditions return to the set point and then this corrective mechanism is switched off. This is how negative feedback mechanism works. Any questions?
No.
Hiba?
No.
Okay. Now rejoin, then we'll continue with kidney failure.